amino acid metabolism. In this session, I'm going to talk about the degradation and the synthesis of the amino acid. At first, I will start with what happened to the carbon skeleton of the amino acid. And as I mentioned, some amino acid can be produced by some other amino acid and some precursors, and they consider as a non-essential amino acid. And also, I reviewed the synthesis of this non-essential amino acid, and at the end, I will review the conversion of the amino acid to some specialized products like histamine, nitric oxide, and some hormones. And I will introduce some nitrogen-containing compounds. As I mentioned, the catabolism of the amino acid is involved in the removal of the nitrogen. After removal of the nitrogen, the remaining compound known as the alpha-keto acid or carbon skeleton. Carbon skeleton actually is considered to the important intermediates that convert to the several compounds. For example, this pathway may be converted to the seven intermediates, like they participate in glucose or they participate in a lipid biosynthesis, and also maybe produces some ketone bodies, and they produce the energy from Krebs cycle. As you know, we have a 20 amino acids that normally participate in the structure of the amino acid, and they naturally occur in the structure of the proteins. 20 amino acids finally, after removal of the ammonia group, they finally convert to the pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. And also, sometimes they convert to the acetyl-CoA or acetoacetate. These are examples of the ketone bodies, okay? Because of this concept, we can classify amino acid in a glucogenic or ketogenic amino acid. In the next slide, I will introduce. So, some amino acid, according to the nature and according to the R group of, so because, why? Because the difference between amino acid is the R group, is a side chain. So, the difference between amino acid is the R group. So, according to the nature of the side chain or R group of amino acid, for example, some of them produce a pyruvate, some of them produce acetyl-CoA and acetoacetate, and the others produce intermediates of the Krebs cycle. So the Krebs cycle is a part of the catabolism of the carbohydrate or glucose. So for classification of the amino acid according to the fate of their carbon skeleton, we can classify as a glucogenic. Gluco means that they convert maybe to the pyruvate. This one. Alpha-ketoglutarate. So these are intermediate of the Krebs cycle, okay? Or ketogenic. Ketogenic means that the amino acids that their carbon skeleton convert to the acetyl-CoA and acetoacetate. So please remember this name of products, because in the next slide I will ask you, for example, the amino acid X convert to the, for example, fumarate. So what is the classification of the amino acid? This is a glucogenic. And for example, the other amino acid produce acetyl-CoA. So this amino acid is ketogenic, because please remember the name of keto, because it's very similar to the name of the ketone bodies. Okay, about the classification, you should memorize each amino acid is belong to which group, and which of them is essential and which of them is non-essential. So we can classify again amino acid according to the production of the end product or the intermediates of the Krebs cycle, or acetoacetate or acetyl-CoA. As you can see here, we can classify in glucogenic, ketogenic, and both. Okay? So we have some amino acids that are exclusively ketogenic. we have some amino acids that are exclusively glucogenic. And sometimes we have amino acids that are both. So they are all not only glucogenic but also ketogenic. What is important here, we should know that leucine and lysine are the exclusively amino acids that belong to the ketogenic group. So they can only produce some ketone bodies or precursors of the ketone bodies. So glucogenic, about the classification, the amino acid whose catabolism yield the pyruvate or some of the intermediate of the Krebs cycle, succinyl, CoA, fumarate, and other compounds. And these intermediates are the substrate. Why we say glucogenic? Because they are substrate for the gluconeogenesis. What is the end product of gluconeogenesis? No, this is glycolysis.
Glucose.
Glucose. Gluconeogenesis is a reverse direction of the glycolysis. In the glycolysis, glucose finally converted to the pyruvate, but for gluconeogenesis, we reach to the glucose. This is a reverse direction. And it happened inside the liver and the kidney. And about the ketogenesis, as the name implies, they finally produce acetoacetate as one type of ketone bodies, and also mean its precursors like acetyl-CoA or aceto-acetyl-CoA. Okay, what about the catabolism? Remembering the name, this part of my speech contains some points and you have to memorize, okay? For the previous session, we should understand the concept. For example, what was the transamination? What happened to amine? Sometimes it's liberated in a free form, sometimes it's transferred to the alpha keto acid. And these are, in this session, you have to, unfortunately, I do apologize, you have several points you have to memorize, okay? And understanding is more easier, but maybe it's more difficult. Understanding is more easier, but it's more difficult to memorize. But what's important, for example, in the final exam, I ask you, each amino acid actually produces some precursors of the Krebs cycle. For example, I ask you, which of the following amino acids produce oxaloacetate? You should know that asparagine and aspartate. For example, when I were a student, I make some abbreviation with them. You can help yourself. You can make, for example, I don't know, this is G.
Like cola and something?
For example, anything like this. You can form an abbreviation.
For example, oxaloacetate, it has to be.
For example, this is a GAP. Am I right? For alpha ketoglutarate. Okay? But you should memorize the first name, and you should not actually make a mistake. Maybe this is the glutamine or glutamate. Okay? You can form some abbreviation like if you are familiar with the formation of some points to memorize it better. So, for example, asparagine and aspartate produce oxaloacetate, and glutamine, proline, arginine, histidine produce alpha ketoglutarate. In the next slide, I will review for the several times. But this is a brief review of the fate of the carbon skeleton of the amino acid. For example, this list of amino acids produce pyruvate. Phenylalanine and tyrosine produce fumarate. Threonine, methionine, valine, isoleucine produce succinyl-CoA. And finally, this amino acid considered as a ketogenic amino acid and produce acetyl-CoA or aceto-acetyl-CoA. So, in the next slide, I will review some mechanism, okay? Some of the catabolism pathway of amino acid is difficult because they contain several precursors, intermediates. So, during my speech, I will emphasize to the important point to memorize. For example, here, you should know that there is a very close relationship between the catabolism of the asparagine and aspartate. This is a structure of asparagine. If I take amine group from asparagine, what would be the result? Asparagine. What is the name of this enzyme? Asparaginase. Asparaginase. Where I heard this name?
It dissolves, it hydrolyzes asparagine in the liver.
In the urea
Cycle.
So we have asparagine. And also after transamination, what is the full name of this transamination?
Aspartate
transaminase. So what is the end product? Oxaloacetate. And now the amino acid that form alpha ketoglutarate, actually several amino acids are converted to the alpha ketoglutarate by means of intermediates, and this intermediate is a glutamine. So glutamine, proline, asparagine, and histidine, by the mediation of the glutamate, finally after transamination, they result in the formation of the alpha ketoglutarate. About the glutamine, the structure of glutamine is very similar to the asparagine, and the general concept is the same. So glutamine, after glutaminase, convert to the glutamate. And also this is a transaminase, alanine amino transaminase here. Convert to the alpha ketoglutarate. And also sometimes throughout the oxidative deamination, if you remember that important slide, glutamate instead of transamination participates in a glutamate dehydrogenase. Okay? It liberates free ammonia. It doesn't transfer amine. It's liberated free ammonia. Actually, this session is more review. Okay? And there is some new points to memorize.
So this step, the one before, liberates ammonia to the urine. And that one turns it into urea.
Pardon?
That one turns it into urea and then into urine.
Yes.
But this one turns it instantly, that it goes straight up to the urine.
Yes, excellent, exactly. And for example, as I mentioned, some of the amino acid metabolism contain complicated steps, like histidine. As you can see here, I start here. Histidine by the histidinase convert to the, orokinase and other compounds. What is here important? You should know that during the metabolism of the histidine, we have very important intermediates, and the name is, with abbreviation, FIGLO. FIGLO is the N for miminoglutamate. Okay, remembering just the abbreviation would be enough. FIGLO. What is the clinical importance of these intermediates? As you can see here, at the subterminal reaction, we require folic acid, B9 vitamin, for conversion of FIGLO to the glutamate. The name of enzyme is not important, okay? Here, just the name of coenzyme is important. As you can see here, because for this conversion, we require a sufficient amount of acid folic. What happens in a case of deficiency and lack of folic acid?
It stops alpha ketoglutarate from
So what would be the result according to this reaction? Whenever you have a lack of coenzyme, is it possible to continue the procedure? No. What would be the result?
Accumulation of FIGLO.
Substrate. So whenever, no matter, you have a lack of enzyme or a lack of coenzyme, your enzymatic reaction cannot continue and stop. And what would be the result? Accumulation of the substrate. So if as a physician, you want to evaluate the status of the folic acid's level, okay? You should order the measurement of the FIGLO. So whenever your patient has a actually very high concentration of bigelow, this intermediate is closely related to the amount of the level of the folic acid of your blood. So, sometimes we want to evaluate the level of some vitamins. For example, how you can evaluate the vitamin D as a physician. Maybe your patient asks you, I don't know, maybe I have a low amount of vitamin D. As a physician, what do you do?
Vitamin D.
Yes, lab, please measure vitamin D, okay? I mean that sometimes if you want to evaluate the level of some vitamins, you order the direct of the vitamin and the intact form of the vitamin, like vitamin D. But it is not very common, for example, for folic acid. Lab, please measure the folic acid. This is not very common. Instead of folic acid, we should order the measurement, the concentration of the bigelow. Okay? But for example, if you remember, for example, for the thiamine, B1 vitamin, how we can determine the level of vitamin? By transketolase enzyme. Because transketolase enzyme was one of the important enzymes that uses vitamin B1. Okay? So, transketolase, where did you hear this name, transketolase? Transketolase. During the carbohydrate metabolism.
Transketolase.
I wasn't.
Pentose,
I was absent.
phosphate,
Pentose phosphate pathway.
pathway.
Pentose
One
shunt.
of the important enzyme was transketolase, and this enzyme requires vitamin B1. So as a physician, if you want to evaluate, for example, your patient had a beriberi disease. Beriberi is as a result of lack of vitamin B1. So how we can evaluate the lack of vitamin? Please do not order vitamin B1. The activity of transketolase is directly to the amount of vitamin. And here, for the folic acid, is closely related to the amount of the folic acid and bigelow. So folic acid related to the bigelow. Some amino acid convert to the pyruvate. For example, alanine, serine, cysteine, and threonine. And you should consider there is a close relation and interaction between glycine and serine. In the next slide, I will describe it. And about the transamination, I think this is the 10th time I repeat this reaction. Alanine convert to the pyruvate. And following this reaction, we have a coupled reaction by the conversion of the alpha-ketoglutarate and glutamate. I can take a note to remember this reaction, inshallah. And alanine is the major gluconeogenic amino acid. Maybe, I don't know, who actually teach the carbohydrate metabolism, Dr. Dashi or Dr. Panani? Dr. Dashi. I think you are familiar with some precursors of the gluconeogenesis.
Yes.
Okay? One of the most important of the precursors of the gluconeogenesis to produce glucose is alanine. Okay? This amino acid deserves a special attention. We have an interconversion of the serine and glycine by the oxidation. Again here, you require acid folic. The full name is not important, just know that for the conversion of the glycine to serine, we require acid folic. But as you can see here, glycine has two fates. Sometimes digests to the CO2 and ammonia, and sometimes it converts to the serine. So as you can see here, please tell me, serine is an essential amino acid or non-essential?
Essential.
Essential serine?
Non-essential, because you can make it from glycine.
It's non-essential. Why?
Because it can be produced by the body.
Excellent. Because we have a precursor, but we have a condition. Please consider this point. We have some condition for some kind of non-essential amino acid. What is the condition? Whenever you have a sufficient amount of glycine, your body can produce a serine. if you have a lack of glycine, if you have a deficiency of glycine, can your body produce a serine?
No.
No. So in this situation, I mean the lack of glycine, serine also is an essential amino acid. Okay? In any condition that amino acid convert to the other amino acid, for example, phenylalanine convert to the tyrosine.
Tyrosine.
Okay? So tyrosine is a non-essential.
But
If you have a
deficiency.
sufficient amount of phenylalanine. In a lack of phenylalanine, not
Tyrosine.
only phenylalanine is essential, but also tyrosine is essential. So we should consider this point. And following the conversion to the glycine to the serine, it's converted to the pyruvate. And phenylalanine and tyrosine convert to the fumarate. So this is actually biosynthesis of the tyrosine from phenylalanine. As I mentioned, tyrosine, because it can synthesize from phenylalanine as a precursor, considered as a non-essential amino acid. This hydroxylation is very important, and this is not a simple enzyme. This is a very large and complex enzyme, and require oxygen and require a special coenzyme. And the name of coenzyme for this enzyme is a tetrahydrobiopterin. So if I ask you, tetrahydrobiopterin is an example of coenzyme, is required for which of the following enzyme? We should know that for the hydroxylation of the phenylalanine, as you can see here, what is the difference between phenylalanine and tyrosine structure?
Just one O molecule.
OH molecule or
OH.
hydroxy group. So whenever we say hydroxylase, it means that you added hydroxyl. Okay? After hydroxylation, after adding hydroxyl, phenylalanine converts to the tyrosine. Have you ever heard the name of phenylketonuria? You know, maybe you think that why I have to memorize all these details. Because as I work in a Shariati hospital of the Tehran, we have a special clinic of the metabolic disorder. And when I were a student, I exactly think like you. And I all the time complain why I have to memorize all these details. But whenever I refer to that center for some research, I actually encounter with the large cases, newborn cases, child who suffer from metabolic disorder of the amino acid. And unfortunately, nowadays, these disorders are very common. You know, I think this is a very rare situation, but unfortunately, many newborns have these disorders. Okay, so as a physician, okay, you should know that what is the principle of this disorder. For example, you have maybe in the future you have several cases of this disease, so you should be familiar with the actually the origin of this disease and also with the basis of this disease. Please do not actually complain about why I bother myself to memorize these points, because you have to remember what is the origin to have a best treatment for your patient. For example, for the phenylketonuria, unfortunately, it is very common, especially inside Iran and other countries. What happened here? This is the most common inborn error of the amino acid. This is very common, okay? And what is the reason it can cause? By the deficiency of the phenylalanine hydroxylase. So, what is the characteristic? So, as a physician, you should know that this enzyme is defective. So, whenever we have a deficiency of enzyme, what happens?
The reaction doesn't continue.
Yes, and so the accumulation of the
Substrate.
So in PKU patient, they have a high amount of the phenylalanine. So phenylalanine accumulates and convert it to the other metabolite, that these metabolites are toxic. And we have a hyperphenylalaninемia, and not only we have an accumulation, but also we have a deficiency of tyrosine because there is no precursor. So you have to provide a tyrosine. So, because as I mentioned, if you don't have any information about the pathway that produces tyrosine, maybe you think that, okay, my patient has a hydroxylate, have a phenylketonuria. Okay, what happened? I don't know. But you are different from other people. You know that phenylalanine can result in can actually cause by the deficiency of the enzyme that converts tyrosine. So you should prescribe a tyrosine as a supplement. Not only as a supplement, but because the patient cannot digest the phenylalanine. They have accumulated of phenylalanine. You should ask them, please limit the intake of any food that contains phenylalanine. Okay? We have some, for example, materials and foods that have the lowest amount of phenylalanine. So you should restrict the diet of your patient because you know that phenylalanine is important for the pathogenesis of this disease. As you can see here, this enzyme is deficient in the phenylketonuria.
Does it, this disease, does it also happen because of the coenzyme deficiency?
Sometimes. You have a different type of phenylketonuria, but most of them are as a result of the deficiency of the enzyme. And also, if you have it, it's very clear that if you have a deficiency of the coenzyme, this enzymatic reaction stops here. Okay, what happened in the normal and in the disease? In the normal, phenylalanine converts to the tyrosine. And this tyrosine is very important amino acid. If you didn't supply, what happened?
Glucogenic
You
and
have
ketogenic.
a lack of tissue proteins because tyrosine is a precursor, is a building block, is a beaker of the structure of the protein. Not only it happened, but also tyrosine can convert to the melanin. What is the melanin?
Skin
Pigment of the skin. So they have a fair hair. They have a very, very white skin, abnormal. Okay?
Albinism? Albinism?
Yes. In the next slide, I will introduce this disorder. You are familiar. In which course you will be familiar with these disorders? Where you heard these names?
I heard because I had a friend who was an albino.
Because I think in other courses other than biochemistry, some professors introduce these disorders.
I think that is last course on biochemistry.
Maybe.
It's
And
biochemistry
also,
too.
because you have several professors, maybe other professors introduce these disorders too. So, tyrosine, why is important? Because this is a source of amino acids for proteins, this is a source of precursors for melanin and also catecholamines like epinephrine, norepinephrine, dopamines. So, in a phenylketonuria, because the conversion of the phenylalanine is blocked and stopped here, there is no catecholamine, melanin, and also tissue proteins. And also, tyrosine, following the catabolism, convert to the fumarate and acetoacetate. So, according to this procedure, phenylalanine and tyrosine are glucogenic or ketogenic.
Glucogenic and
The
ketogenic.
degradation produces fumarate and acetoacetate.
So they're both. They're
both.
glucogenic and ketogenic.
Why? Because
They
this
produce
is
both
glucogenic because of fumarate and ketogenic because of excellent. What is the characteristic? As I mentioned, the substrate concentration is very high and it accumulates in the plasma and in the urine. And not only phenylalanine, but also we have some metabolite that derive from alanine, phenylalanine, like phenylpyruvate and other metabolites accumulate. And because of this metabolite, the urine of the PKU has a mostly other. You know, many years ago, there is no specific method to measure the concentration, for example, phenylalanine, to determine the activity of phenylalanine hydroxylase. How they can determine the PKU? They just smell. For example, many years ago, how people determine the occurrence of diabetes? They taste the urine.
It's sugary.
It's sweet.
It's, yeah.
Many years ago, it's disgusting, but that was true. For example, 100 years ago, there is no specific kit for the measurement of the glucose. For example, they were very thirsty, they actually go to the doctor for it several times, and the physician wants to evaluate the diabetes, they just taste, okay? So, for this disorder, for example, many years ago, they understood that the urine of this patient has a mousy odor. And what are the symptoms? They have some intellectual disability, developmental delay, and some problems regarding the nervous systems and seizures. As I mentioned, because one of the precursors of the melanin is this is a precursor of the melanin, they have an abnormal light skin. They have an abnormal blue eye. Okay, this is not normal. They have an abnormal blue eye because of lack of melanin. Okay. Blue eye. Do you want to have a blue eye? Light skin, blue eye. At the first glance, it seems that it's very beautiful. Maybe at the first glance, think that most of the European people have a PKU. But this is abnormal, okay? No, he is normal.
So there's.
Okay, about the catabolism of the tyrosine. The catabolism of tyrosine is very important because here we have another metabolic disorder, and again this metabolic disorder is very common, unfortunately. What happened after transamination? Just know that the transamination. Finally, we have a hemogen. Just the name intermediate is important. So you should know that during the catabolism of the tyrosine, we have an intermediate called hemogentisate. Whenever you have a deficiency of this enzyme, and the enzyme is a hemogentisate deoxygenase, we have the accumulation of hemogentisate. So what is important in this slide? We should know that intermediate of the tyrosine is a hemogentisate. And what is the digesting enzyme of this metabolite? The name of this enzyme is important. Because why? Because whenever we have a deficiency of hemogentisate deoxygenase, again, we have a lack of enzyme. What is the result? Accumulation of the substrate. What is the substrate? Hemogentisate, and it accumulates especially into the urine and other parts of the body, like this. This is a sample that's taken from the patient who suffer from alkaptonuria. Alkaptonuria is a disorder and is a genetic disorder. In this situation, this enzyme is deficient. What happens, for example, if you take a urine from your patient and after 15 minutes, it can show a small pigment, dark pigment inside the urine, okay? And after 2 hours, the urine is entirely black, okay? So if you want to determine and distinguish the occurrence of the alkaptonuria, please, lab, please take a urine from the patient. And whenever you take for the 15 minutes, after 15 minutes, urine start to convert to the dark pigment. And actually, finally, after 2 hours, it actually completely is black. Not only the hemogentisate accumulate into the urine, but also this black pigment deposits on the intervertebral these of the vertebrate. So they have some movement problem because they accumulate in a nervous system. So these are the symptoms of the alkaptonuria. And as I mentioned, you have several cases, and in the future you will be encountered with these cases. And about what is it?
Albinism.
Okay, this is a deficiency of melanin. As you can see, is it beautiful? No. There is no melanin. This is no beautiful actually. As a result of some enzymes that are responsible to the conversion of the tyrosine to the melanin pigments, they have a hypopigmentation, they have some vision disorder, and they have a photophobia. Photophobia, it means that
Fear of
fear of the light. And they have it increased, especially they are in a dangerous of the skin cancer. Because our melanin protects us from the dangerous light waves and lengths of the sun, especially UV light. Some amino acids convert to the succinyl coenzyme A, like valine, methionine, isoleucine, and threonine. Between these amino acids, methionine is very important. Why? Because in some metabolic pathway, you want to produce a specific compound.
Cysteine?
Methionine actually. Because in some, as I mentioned, in some metabolic pathways, if you want to produce new compounds, you have to transfer a methyl group. What is the donor of the methyl group? For example, this is the S-adenosyl methionine. Okay? This is a very important donor, especially in many enzymatic reactions, and serves as a precursor of the methyl transfer reaction. So, following the condensation between ATP and methionine, we have a SAM. SAM, if I ask you, what is the function of SAM? It is a precursor of the methyl group. So in the next slide, we have some enzymatic pathway that requires methyl. So methionine provided this methyl group. And about the threonine pathway, threonine has two phases. Sometimes it's converted to the succinyl-CoA and sometimes converted to the pyruvate. So it is a ketogenic, glucogenic, or both?
No, just keto.
Glucogenic.
Keto?
It's both ketogenic and glucogenic because pyruvate and
Succinyl-CoA is a precursor of
It's in the TCA cycle.
Is there any acetoacetate? No. Is there any precursors of the ketone bodies? No. Both of the pyruvate and succinyl-CoA considered as a glucogenic. Okay? So threonine is a glucogenic. Maybe you make a mistake from CoA. Okay? CoA is not important. Okay? Because intentionally I mentioned this one for your final exam. Please read full name. Succinyl-CoA is not ketogenesis. Okay? This is the precursors of the Krebs cycle. So, for your final exam, if you immediately choose the item, you can make a mistake. Okay? So, please read the complete name. About the catabolism of the branching amino acid is very important, but the detail is not important. Okay? Because we have several compounds. What is important here, you should just know the name of branch chain amino acid. According to the specific structure of the R chain, we classify them according to the specific branch form of R chain. What are these names? What are the names of? Valine, isoleucine, and leucine. After aminotransferase, they produce some compounds you don't supposed to memorize. And also after dehydrogenation, they produce other compounds and convert finally to these end products.
So then what exactly should we remember from this slide?
I will tell you. The name of branch chain amino acid, valine, isoleucine, and leucine. So, I ask you, leucine is a ketogenic amino acid or glucogenic amino acid, and why? Keto, why?
It's keto because they make acetoacetate.
Because of the formation of acetoacetate. Okay. What about valine? What about valine?
It's both glucogenic and keto.
It's glucogenic
Only glucose.
Why?
because they make succinyl CoA.
But it also
This is the end product. Please consider this pathway. Valine finally reached just succinyl CoA. And succinyl CoA is the intermediate of the TCA cycle. What about isoleucine?
ketogenic.
Ketogenic.
Why ketogenic?
Because they make
Because
acetyl
acetyl
CoA.
CoA.
What about the glucogenic?
Because it ends at succinyl CoA.
Excellent. So you should consider, at the beginning of the slide, I mentioned this point. Leucine is the exclusively ketogenic.
Ketogenic.
Valine is only glucogenic. And what about isoleucine? Ketogenic and glucogenic. Clear? So this point is important. The name of branching amino acid and the fate of each amino acid. You should know that leucine is exclusively ketogenic.
Ketogenic.
Isoleucine is
Ketogenic and both glucogenic.
what about valine? Only
Glucogenic.
glucogenic. Why? Because it produces only
Succinyl.
a precursor of the Krebs cycle. And this is actually complex of the dehydrogenase. In a phosphorylated form is inactive, and in a dephosphorylated form is not active. Only this part and the end product is important for you. Okay? About the why I mentioned that metabolism of the branched amino acid is important, because again you have a very common genetic disorder, and this is a MSUD. And for the future also you have this case. You have this newborn case, okay? And maybe you have a children that the physician cannot distinguish at the beginning of the birth, okay? So you have several cases, unfortunately. So you should be familiar with the basic of this disorder. As you can see here, during the catabolism of the amino acid that belong to the branching amino acid, we have a dehydrogenase. And this is a very large complex, and it is very important. So, during the MSUD, maple syrup urine disease, again, the taste is like a maple syrup, okay? The deficiency of this dehydrogenase is present. So, as a deficiency of enzyme, again, you have accumulation of the substrate. So, you have accumulation of the alpha keto acids here. Whenever you have a deficiency of enzyme, these alpha keto acids, why we say these are alpha keto acids?
Because
This is a aminotransferase. All the time, whenever you have an aminotransferase that acts on the amino acid, what would be the result? Alpha keto acid. So whenever you have a deficiency of dehydrogenase, these alpha keto acid accumulate. But the name is not important. Just know that these alpha keto acids accumulate and can result in some problems, like feeding problems, like vomiting. They have a low amount of water, their body are dehydrated, and they have a very acidosis. Why the acidosis?
Because of high keto acids?
Excellent. Because of the accumulation of alpha keto acids. And these are acids. So they change the pH to the acidic environment. And also about the characteristic, this is the other of the urine again, because all of these metabolites exert to the urine. So they have a mental retardation, and if it is untreated, it can result in a death. About the synthesis of amino acids, these are essential and non-essential amino acids. For example, I mentioned that alanine is a non-essential. And again, for the 1,000 time, pyruvate convert to the alanine. So alanine is a non-essential. Oxaloacetate can form an aspartate. So aspartate is a non-essential. And alpha-ketoglutarate convert to the glutamate. And also, if we want to produce a glutamine, our body uses glutamine synthetase by the accumulation of the ammonia. So this is a major mechanism of the ammonia in a non-toxic form. So glutamine, if you remember, this is a non-toxic form of the ammonia. And again, we have an interrelation between aspartate convert to the aspargine. Who donate this amine group? Glutamate. So whenever aspartate want to convert to the aspargine, we require one amine group. Who is the donor? Glutamine. And about the proline, if I ask you during this slide, this is just this, the title is important. The precursors of the proline is glutamate.
And it's the other way around as well?
Pardon?
It's the other way around as well? Glutamate
No. Just the precursors. What is the intermediate, the name of intermediate, the name of the coenzyme, enzyme, you don't suppose. Because there it is full of points, okay? And for the physician, it is not important. You should just know the principle of the catabolism, the principle of many diseases, you know? Okay, about the serine biosynthesis, serine can, because I'm talking about the synthesis of amino acid, we have a two pathway. One of them from the precursors of the glycolysis. This is, if you remember, this is the precursors of the glycolysis. Not only it continues to convert to the pyruvate, in other pathway it can produce finally serine. Again, the details is not important. You should just know that how our body produces serine. Sometimes from the intermediate of the glycolysis, the name is important, and sometimes because we have the interconnection between glycine and serine, not only serine can produce a glycine, but also glycine can produce a serine, because this is a reversible reaction. And for example, about the cysteine, we should know that methionine is a precursor of the cysteine. But sometimes during this procedure, we produce the homocysteine. This point is not important. Maybe this point is important for you because you are a physician. Sometimes you want to order some markers regarding to the cardiovascular disease. You should know that if your patient has an abnormal level of homocysteine, homocysteine is a specific amino acid that actually does not participate in the structure of protein. What is clinically important? You should know that homocysteine is related to the atherosclerosis. Are you familiar with atherosclerosis? In Arab, tasallub al-sharaiyah.
Yes.
Yes? Am I right? Yes? I gradually understand. I learn some. هل فهمت ذلك؟ Okay, where are you from? India?
Yeah.
I do apologize because most of the international students, maybe more than 90% of the international students in Tehran are Arab, and they are very actually happy when I use some Arab words, okay? Because this is very easy for understand for them. For example, I think in Iran only I have one student from South Korea.
South Korea?
Yes.
I'm one from Afghanistan.
Afghanistan?
Yeah.
You cannot understand Arab language, yeah?
No.
And also we have a few students, yes, less than 10 students from Afghanistan, and most of them, more than 90% are Arab. So I have to, because I have to actually go to the subtain, so I have to learn some Arab words, okay? So I do apologize if I use some Arab words. So تصلب الشرايين, okay? Atherosclerosis, hypertension, okay? And the blood clotting is related to the accumulation of the homocysteine. So homocysteine is a biomarker of the CVD or cardiovascular disease. So as a physician, if you are suspected to the atherosclerosis, high blood pressure, you can order the measurement of the homocysteine. But for the amino acid metabolism, you should know that what is the precursor of the cysteine? Methionine. And this is a key concept. This is for more information. As I mentioned, only a few slides remain. As I mentioned, not only amino acid considered as a building block of the proteins, but also they can convert to some other compounds that contain nitrogen. For example, some hormones, some precursors of the nucleic acid. Okay? So these are nitrogen containing compounds. What are the example? Catecholamines, like dopamine, epinephrine, and norepinephrine. Histamine, serotonin. What is serotonin? Happiness hormone. Okay? And creatine and nitric oxide. Have you ever heard the name of catecholamines?
Yeah, just
Epinephrine. Norepinephrine. So these are categorized as a catecholamines. They synthesize mainly by the brain as a neurotransmitter. They transfer the neuron signals. And norepinephrine is also synthesized in an adrenal medulla. What are the functions? Norepinephrine and epinephrine, they are hormone regulators of the carbohydrate, lipid metabolism. They accelerate, For example, if you're in a hurry, our body produces these catecholamines. And so it means that we require enough energy. So they increase the rate of the catabolism of the carbohydrate to produce more energy. And also they release from adrenal medulla in response. For example, if you suddenly frighten, okay? Suddenly our body produces catecholamines, okay? And for example, if you are in a vigorous exercise, actually unexpected, severe exercise, or in a winter, in a severe cold, and also in a low level of blood glucose, our body wants to compensate the lack of glucose, so it accelerates the degradation of the glucose and teaches you to produce more energy, to be ready for the fright, for the exercise, for the cold. About catecholamine biosynthesis, the precursor is tyrosine. You should know this pathway is very, very important. Why? Because I mentioned some disorder of this pathway. Whenever you hydroxylate your tyrosine, you have a DOPA compound. And after removing of the carboxyl by means of decarboxylase, you have a dopamine. Have you ever heard the name of Parkinson?
Yes.
Yes. This is a neurodegenerative movement disorder, especially in old patients. Okay? What is the reason of this patient?
Dopamine deficiency.
Too much
Loss
dopamine.
of dopamine producing cells inside the brain. So, as a physician, what would be the best treatment?
Drugs.
Drugs.
Weed.
Which drugs?
Dopamine.
Dopamine.
Excellent. Because the brain cannot produce enough amount of dopamine to control the movement, so you can prescribe dopamine as a supplement.
That's like type of depression.
Depression. Because most of these catecholamines can affect the mood. Also can affect some neurodegenerative disease, because most of them are originated from the different elevation and altered concentration of these catecholamines not only are important for some neurodegenerative disease, Parkinson's, Alzheimer's disease, but also they can affect your mood. Okay, if you have a depression, if you are in a full of a stress situation, some physician prescribe. But for a normal patient, please do not actually suggest these drugs because they are very, not as strong because the patient habit to take these drugs, like addict persons.
Yes.
You can advise them to change their lifestyle. Some patients do not require to these drugs. Okay, if they change their lifestyles, they can control many mental disorders. They can change their mood. It is not required because any drugs have a side effect.
Yes.
Okay, so if you can control by the exercise, by the change in your lifestyle, why it's necessary to prescribe a drug. But sometimes, for example, for Parkinson's, we know that our brain cannot produce this neurotransmitter, so you have to prescribe the drug.
This is specific.
Yes, specifically can affect the movement of the organs.
I just have a question. Tyrosine to dihydroxyphenylalanine. What is BH4 and then O2? Like what is
Tetrahydrobiopterin. Because in the previous slide, we have a phenylalanine hydroxylase, and at that slide I wrote this abbreviation. Again here, most of the time, especially when you have a hydroxylation of amino acid, tetrahydrobiopterin is required.
So, which means that this and as well as tyrosine hydroxylase, they are both needed.
Both needed to the tetrahydrobiopterin as a coenzyme.
And they need oxygen too?
Yes, as a source of, you add a hydroxyl, but the source of hydroxyl is the oxygen. Because inside the structure you have a hydrogen. Whenever you add the oxygen atom, the combine of oxygen and hydrogen is hydroxyl group.
And from dopamine to norepinephrine, vitamin C and copper,
Yes.
what are those?
Yes. I continue here. I reach here, Parkinson's disease, actually as I mentioned, as a result of the deficiency of this dopamine, but we do not stop here. We continue this procedure. By another hydroxylation, you produce the norepinephrine, and after methyltransferation, what is the source of methyl?
SAM.
S-adenosine or SAM. It provides a methyl to produce epinephrine, or the other name is adrenaline. So dopamine, norepinephrine, and epinephrine, all of them are neurotransmitters. Question?
Is this cycle happen in the brain?
Cycle?
Does this happen in the brain?
Yes. And also, in which part of the body? Adrenal,
medulla.
medulla. And about the metabolism, what is important here? The details is not important. You should just know that you have a monoamine oxidase and methyltransferation for the catabolism of the catecholamine. These metabolites is important again because unfortunately you have this case, pheochromocytoma. This is another disease. As a physician, you should be familiar with the basic of this disease. Pheochromocytoma, in Farsi pheochromocytoma, is a tumor of the adrenal, okay? By the excessive production of the catecholamine. But normally, as a physician, we order the measurement of the catecholamines. Lab, please, epinephrine, norepinephrine, no. Most of the time, they order the measurement of the exertion metabolite. What is the exertion metabolite? VMA is very common between the physicians. They order VMA and HVA. This is homo-vanillic acid and vanillyl mendelic acid. So as a physician, if you are suspected to some malignancies of the adrenal gland, so you should not order order the measurement of the catecholamine. You should order the measurement of this urinary exertion of these metabolites. And if they are high, so you should start the chemotherapy and radiotherapy of the adrenal gland. It means that there is some malignancy of the adrenal gland.
So we just need to know that MAO and COMT, they help norepinephrine turn into VMA.
And also, what's important? The name of
the
metabolite that exert into the urine. So they measure not in the serum, they measure in the urine. So as you understand in this session, urine is a very important sample for the evaluation of the amino acid metabolic disorders. Okay? And about histamine, I think you're familiar with these molecules. Okay? It can result in itching and allergy. What is the source of histamine?
Mast cells.
Histamine. After decarboxylation, histamine released by the mast cells, and it can act as a vasodilator. It can decrease the blood vasodilation, and is a mediator of some response, especially in inflammation. Okay. And about the serotonin, this is a happiness hormone. Okay. For example, any situation that make you happy, okay, can result in even eating ice cream, okay? Any situation that make you happy can result in a secretion of the serotonin. What is the precursor?
Tryptophan.
And so the largest amount of this hormone is found in intestinal mucosa and a smaller amount in a CNS. What is the physiological role of this hormone? This is a pain perception, regulation of sleep, appetite, and it can control, in a simple word, it can control your mood. And this is a production of the creatine and creatinine. This is a deposal form of the creatine. Creatine, where we can find the creatine?
Nails in the
skin as well.
Nails.
No.
And also
Muscle.
muscle.
Creatine kinase in muscles.
In the muscles, do you remember the creatine kinase reaction?
Yes.
We have a phosphorylated form of creatine as a source of phosphor to actually convert ADP to the ATP. So, how we can produce this important molecule? By the condensation of the arginine. This slide is very important. But you should know that if I ask you which amino acid are the precursors of the creatine, you should know that arginine and glycine. After condensation of arginine and glycine, we have a guanidino acetate. And again, we require methyl group, and some participate in this reaction and produce the phosphocreatine. Which organ are participate? The first reaction take place inside the kidney, and the second, because guanidino acetate sent to the liver, and in the liver we produce the phosphocreatine. And the same inside the muscles. And sometimes when we have the excess amount of creatine, it converts to the creatinine in the urine. And if you require ATP, phosphocreatine release its phosphate and donate to the ADP and produce the creatine. And the last important nitrogen containing compound is a nitric oxide, and sometimes it's considered as an inflammatory compound, and it contains nitrogen, and it has several roles. But what is important here, we should just know that the arginine is a precursor of the nitric oxide. Okay, this is a review of the conversion of the amino acid. So, tyrosine is a precursor of dopamine, norepinephrine, epinephrine. All of them are catecholamines. And also they produce melanin. So whenever you have a deficiency of tyrosine, you have a hypopigmentation, you have a hair, and also in the structure of the thyroid hormones, you have a tyrosine. After adding 3 iodine atoms to the tyrosine, you have a T3 hormone. Okay? If you add 4 iodine,
T3,
T4. So T4 and T3 are the important thyroid gland hormones. So it precursors is the tyrosine.
Can we also say that the precursor is phenylalanine?
Yes, because if you provide the tyrosine, there is no necessity to the phenylalanine. Okay, but if you don't provide the excess amount of the tyrosine inside the body, at first phenylalanine convert to the tyrosine and tyrosine actually. But if you, for example, if I ask you the precursor of the thyroid hormone, it's better to write a tyrosine, not phenylalanine. And this is a histamine that's derived from histidine, tryptophan, converted to the serotonin, and also melatonin. Melatonin, please do not make a mistake. Melanin and melatonin. Melatonin is
For
a
sleeping.
pineal gland hormones that regulate the
sleep cycle.
Excellent. And also about methionine, as I mentioned, provide a SAM as a methyl donor, methyl transfer compound for production of the creatine and epinephrine. And about glutamate, you have a GABA. These are the example of the neurotransmitters. And also cysteine can produce glutathione is the antioxidant compound, and glycine also produces glutathione, and glycine also participates in the production of the creatine, and arginine can participate in the production of the creatine and also nitric oxide. Oh, is there any question?
For glycine, we can also make serine.
For lysine? Glycine?
Yeah. We can make serine as well.
Serine, yes. These are the not amino acids. As you can see here, these are not amino acids. These are other nitrogen compounding, nitrogen containing compounds, like all the time in the countries. These are, for example, hormones. These are neurotransmitters. These are some molecules that can control some aspect of the metabolism. As you can see here, between these compounds, there is no any amino acids. You know, for example, if I can draw, it's a very complex. Phenylalanine to tyrosine, glycine to the serine. This is the interconversion between amino acids. But here, this is a conversion of amino acid to some specialized nitrogen-containing compound. And as you know, some of them is a pigment, some of them is a hormone, some of them is an antioxidant. Okay, some of them is a reservoir of the energy. Many thanks for your attention. It's finished. Alhamdulillah, it's finished.